this is the second exciting seminar brought to you by Exciting Instruments. And I'm going to tell you a little bit about our first exciting instrument in a moment. But as I say, Thorsten Hugel will be delivering this really interesting seminar on 3-color single molecule FRET, a game changer, ? And he has just been delayed by a train by 2 hours. So he's not going to be 2 hours late to the seminar. You'll be glad to know he'll be with us in about 5 minutes. But until he gets here, I'll just give you a bit of an overview of other things that we're up to. This is the second in our seminar series. We're going to have 2 further seminars already planned with Professor Victoria Berkedel and Professor Achilles Kapanidis, giving some great talks coming up, and you'll be receiving invites to those. The idea is these are every couple of months, this sort of time. So hopefully you enjoyed the first one from Shimon, and you're going to enjoy this one from Thorsten. So I just, before Thorsten gets here, give you a brief intro to our Exciting Instruments and what we're all about, and our instrument that we are now in production of the first few. So the real background to the company is the driving force for what we're trying to do is to really democratize single molecule spectroscopic methods. I really mean that by many of you are aware of the smFBox work that my lab did in sort of making a build your own single molecule FRET confocal instruments. And what we've done is try to make that available through the company in a slightly different form factor, but the same idea. We're backed by open science, and to make it available for people who can't build it themselves, basically, to make single molecule methods available to the widest possible user base. The company itself is a joint venture between the spin out from my lab and a local tech firm who bring a wealth of software knowledge, which give us a cloud connected platform. So it means you get your data, you can analyze your data on the cloud, and you can see live updates of how your experiment's going on your phone as the experiment's progressing, which is kind of cool. And part of making things easy to use at the end of the day is software, right? It's all well and good having a nice piece of hardware, a good piece of kit, but the software is integral to usability. And for us, democratization is about 2 things. It's about usability and it's about affordability. And we really tried to hit both of those barriers to bring those down to allow people to use our kit. And we'd really like this, if people have got a fluorimeter in their lab, they really ought to have an EI flex. Why do an experiment on the ensemble level or with an ensemble average when you could do it at the single molecule level? You guys all know why we do single molecule experiments, so I don't really need to go over this, but if you're at a basketball game, you've got the players, very few of them, you've got the spectators, many more of them, us mere mortals. Look at him in comparison to the giants, right? I mean, it's crazy, but the average is really misleading, and so it's really important that we can go beyond that ensemble average, and that's absolutely what our instruments are able to do. They're able to allow you to interrogate single molecules. So that's the single molecule FRET box, as we call it, the smFBox here. And all of the same optical and software arrangements that work here. Hi, Thorsten. Take a minute, don't worry, just breathe for a few minutes. All the same optical and hardware elements are incorporated into the new Flex, which includes alternating laser excitation to give you access to donor photons, acceptor photons from FRET, but also directly exciting the acceptor to show that it's definitely there. And obviously all of that, the way we built that is published in the Nature Commons paper that we have there. One of the advantages of the alternating laser excitation is the ability to measure absolute distances with FRET, which we've used and Thorsten himself has pioneered to great effect for actually the study of biomolecular complex determination, and in Thorsten's work on HSP90, looking at the actual dynamic structures adopted by the HSP90 homodimer, some wonderful work over many years there. And also, in fact, work headed up by Thorsten of which I contributed a bit, where we did this study where we sent out these different oligos to sort of 25 labs around the world, and Thorsten didn't tell us where he'd put the dyes, and which was a brilliant blind study that showed using these accurate FRET methods, you can actually measure precise distances if you do all of the corrections correctly. And this is just a plot showing from the Smurf box data. The data shown in grey is from the Smurf box, and the red crosses are the FRET efficiencies that were achieved in the blind study by all of the other labs. So you can see that our hardware does really nicely, just as well as everybody else's, and everyone does well, which is great. And so that's great. So that's the Flex in its all its glory, and it's a very modular system. You can use single molecule FRET with it. You can do FCS with it. The nice thing is it goes on a bench top. It's the class 1 laser product. It has a very small footprint. So in fact, one of our first customers is actually putting it in a glove box to do some work in an anaerobic environment. So that's exciting. Yes, absolutely, Sonia, in a minute. That's a good idea. So yeah, that's the Flex. It's we've got a number of arrangements available. This, for example, is a 2-color 2 different excitation units that you can switch in and out, and that's another version of the software and the hardware that we've got. And the idea is this becomes very modular, so you can customize it as you go. You can increase the number of detectors, and I'm happy to talk to anyone in more detail about the exact capabilities of the different things. In fact, if you chat to myself or to Chris Saywell, who's on the call, drop him an email at info@excitinginstruments.com. We'd be very happy to set up a more detailed call to discuss all of the different things. Brilliant. So I think, Thorsten, are you about ready, or would you like another minute? No, you're good. Excellent. I will just hand over to Sonia for a moment. Sonia, are you able to unmute your mic? Do you have that power? You do. Excellent. And Sonia has been busy organising the satellite meeting for the methods and applications of fluorescence, which is going to focus on FRET. And so I'll just hand over briefly to Sonia to give that a bit of information about that. Over to you, Sonia.
Yes, excellent. Thanks a lot, Tim, that we can briefly squeeze in a little bit of a commercial on something very non-commercial, a community-organized FRET satellite meeting, which is linked to this MAF conference in Gothenburg in September 2022, this year. So if you haven't heard about the MAF 2022, then please look it up. It's happening on the 11th till the 14th of September. And right after, so from the 14th till the 15th, we have added 3 more sessions on different FRET specific topics. So the MAF conference is on fluorescence in general, and this added FRET satellite workshop will be specifically on FRET, biosensing, ensemble methods, but also single molecule, in solution, or surface immobilized, everything covered. And we're still inviting abstracts for talks by early career researchers. Please send them to either me or Don Lamb or Nico Hildebrand by June 5th. That is the deadline for these abstracts for early career researcher talks on FRET. Yeah, thanks.
Brilliant, thanks, Sonia. So with that, we'll crack on with our main speaker. Thorsten, are you able to share your screen?
I will check. It looks good.
Brilliant. That looks great. So I first interacted with Thorsten a very long time ago now. It certainly feels that way in a good way. When we were during this blind FRET study on the what we come to call the nature method standards in our lab, I guess these DNA standards where we get accurate FRET from them. But Thorsten has been a an absolute powerhouse in pushing forward the capabilities of single molecule FRET in many different directions. He runs a great lab of which FRET is one part, alongside AFM and many other interests of his. But I'm really excited about this talk today, where he's going to tell us about 3-color single molecule FRET, a game changer. Over to you, Thorsten.
So thank you very much, Tim, and also 2 exciting instruments for having me in this seminar series. And sorry that I was a little bit late, but I'm sure Tim did a good entertainment in this time. So I had a 2-hour delay on a direct connection. I mean, the German railway is not good, but this I never encountered before. But so yeah, just made it now. Okay, yeah, as Tim announced, I will talk about 3-color single molecule FRET, also a little bit on structure determination and live cell imaging. So I connect to the first talk in this series by Shimon Weiss, who gave us a little historic overview, but also started to tell in which direction single molecule FRET or also fluorescence could go. And I tried to also give you a little bit of a hint on that. So why multicolor single molecule FRET? It's a fundamental difference if you go from 2 to 3 colors, because what you can do is you can look at correlations between conformations and interactions. So both of this you can do with 2 colors. So you can look at the opening and closing of HSP90. You can look at the interaction of another molecule with HSP90, but to tell the correlation or the succession of states in the system is very tricky, if not impossible, with only 2 dyes. And I will show you several examples of this. And also allostery is difficult. So this is not impossible with 2 dyes, but it's also difficult. Just to give you also a very quick historical overview, so there are many more publications on 3 or 4 color FRET. I think nothing with 5. But actually not so much if you consider that, as far as I think, the first real single molecule 3-color FRET study was in 2004, so almost 20 years ago. And it's probably hard to find 20 publications on this. So it's really not too much. We will also maybe see during the talk what are the problems there. I mean, very important was then to do a 3-color alternating laser excitation. As for 2 colors, for 3 colors, this is crucial to figure out the fluorophore physics and also to get real distances and things like this. Yeah, then there were some 4-color studies. So this was all turf-based, and recently Lemke and Lamp Lab also brought a 3-color single molecule FRET for confocal microscopy. So in principle, you would have this available now, at least published. So you don't have to make the first journey yourself. Okay, before I tell you a little bit what we can learn from this, I just quickly remind you of the basic system we are investigating now since 15 years or so. It's the HSP90 machinery. It's a homodimer. It has an ATPase in each of these dimers. Is a multi-domain protein, so 3 domains, N terminal, middle, and C domain. It's also biological, important. It's 1 to 2% of the cytosolic mass in a cell, and it's involved in many signal transduction pathways. There's several hundred substrates. They are called clients for HSP90. Especially a big part of the kinases, more than 50%, people say only works with HSP90, are activated or regulated by HSP90. Yeah, and then it's also a nice model system if you look at conformational changes and interactions with other proteins, because it never works on its own. So it works with several co-chaperones and substrates. I just show here a few of them, so 20 about known. The first 3 we work with, SPA1, CDC37, and AHA1. And then there are many clients. We are mainly, so here I will just show about this kinase, the STIA11 here. Good, so this was a very short introduction to the biological part. So now a very quick introduction into the single molecule FRET part. I assume everyone knows what this is. If not, just think of it as a spectroscopic ruler. So you shine light here on this yellow dye. And if the dyes are far apart from each other, you get yellow light, more yellow light. If they are closer together, you get more red light. And from this ratio, you are then able to say something about the distance of these dyes. So what we usually do in turf microscopy is we have a cover slip or a slide, depending on the setup, and then a microfluidic chamber where you then can flow in your molecule, attach it to the surface, very often on a PEG floor, so polyethylene glycol, such that it doesn't stick to the surface unspecifically. Then you have some bioteins on some of the PEGs, you add streptavidin, and then you have a biotin-related protein and attach this to the surface. You then excite this molecule and only the molecules on the surface. That's why you do total internal reflections, because you don't want the background from all the other molecules. And then you detect, you can do this then on the opposite side. You could also do it on the same side, the different colors. Yeah, here I just show it for you with 2, and we will extend this then to 3 and 4 colors. And yeah, so please interrupt me if something is unclear just during the talk. Yeah, and what you then get is basically the 2 colors on, in this case, 1 camera. You could also have it on 2 cameras. So this would be the donor dye, which was yellow here, and this would be the acceptor dye. And you can calibrate then the position. So you would then know, okay, this is the same one than this one. And then you take this dye and look at it over time. Yeah, so take the time trace out of this. So this would be the donor, and this would be the acceptor. And you already see at the beginning, you see more donor, so they're further apart from each other. Then here, sometimes you see more acceptor, they are closer together. Yeah, and at some point, the acceptor bleaches, and then the donor bleaches. Actually, wrong, the donor bleaches, and then here the acceptor bleaches. And this is actually the third signal which you get from alternating laser excitation. So basically, in between, you excite the acceptor for a short time directly to check if the acceptor is still there, because something like this could also be a blinking of the acceptor or dying of the acceptor. But if you see that it's still there in between alternating excitation, then this helps you. Okay, if you then have these signals, you can calculate something that's called a FRET efficiency, which is in principle the intensity of the acceptor normalized to the total intensity. And this can then be converted into a distance, and I will come to this later on. So for now, we just look at the dynamics. So this is basically the main information you can get. You can get the dynamics of these dyes, how they come closer together and apart from each other again, or you can get distance informations or both. Yeah, but here we just say small distance, large distance. Yeah, and as we know our system, this means when it's up here, the HSP 90, the N-terminal domains are closed, and if it's further down, they are open. Good. So now you can try to get the kinetics out of this. Some people say you know all about the protein if you know the full kinetics, the full free energy landscape. So how can we get this out of it? So you could just do a dwell time analysis, but this is not good enough here because you cannot fit this with a single exponential, these dwell times. I think you hardly can do this in any system. So therefore, you have to go to some other analysis. And this are mainly maximum likelihood-based analysis with hidden Markov models. So assuming no memory in this system. And Sonia, who just talked a few minutes ago here, she wrote this software here. This is not the first one of its kind, but it's specialized for single molecule fluorescence. It's a single molecule analysis of complex kinetic sequences. And the nice thing, I mean, you get the rate constants, also from many other software, but you also get some errors, some estimates, which I think is very nice. Yeah, and then you can calculate the free energies and also fluxes. I will come to this also later. And for the idle HSP 90, this we have already done 6, 7, 8 years ago, and you can then get the free energy landscape. And also, I mean, with this, you have the full information, you have the kinetics, we have the thermodynamics. Good. So now we started to think, okay, how is this modified? What happens if you add some other components to the system? And for this, we like to look at the transition matrix that you have here. So we have here basically two FRET states that you see directly in your fluorescence, high and lower. Yeah, this would be here, high FRET, low FRET. And then you have two hidden states. They just come from the kinetics. If you analyze the kinetics of the system, you see some faster transitions and some slower transitions. And from this, then you can assign these four states. They're called 0, 1, 2, 3. And then you can just ask yourself, okay, how often does the system go from state 1 to state 2, or from state 1 to state 0, or from state 1 to state 2, and so on. And then you can make a model. Here for HSP90, I mean, several people have proposed this. We are also that this is a 4-state model. It's kind of accepted. Yeah, if you can accept this model, then you can resimulate the data because you just know now all the rate constants. And then you can compare this and say, okay, this is okay represented. Yeah, I think the ideal HSP90 4-state model in this time regime that we can measure is probably nice. Yeah. So now what happens if we add some components here? Yeah, here a co-chaperone and a substrate, because I mean, this is then the system where biologists start getting interested in. Yeah, HSP90 on its own is boring. Okay, here we still with 2 color FRET. I will come to 3 colors in 2 minutes. And we just look at this opening, closing. So the same coordinate basically than in the other system, just now 2 more components there. And what you see, it really looks different. I mean, this is nice. So there is some binding, some interaction here. Yeah, but now you start with this 4-state model and yeah, with the rate constant, and you see, I mean, I would say this is not well represented. Also, you can define such a base information criteria, and this looks good here. Yeah, so basically, yeah, mathematics would tell you this is not a too bad model. But the thing is, and this is the problem with these maximum likelihood methods, I mean, they always give you the most likely result given the input. Yeah. And we have input here that we have these 4 states. So this is a problem, and I come to this in a few minutes. Good, and now, and this was without ATP, and then we added ATP, and we even then see a nice change. So one could now say, okay, this is better represented by this 4-state model, but this might also be by chance, yeah, because why should it not be without ATP and then be good with ATP? So this is like a little bit of a caveat, yeah, I mean, you have to be really careful. But nicely we see that ATP has an effect. And so then we ask, is there now some directionality or a cycle? Because this would then really be a molecular machine that basically goes in a certain direction. And this is something that many people proposed for HSP90. And we could not show this for HSP90 on its own. And we were hoping to see this, that we see this if we have clients and co-chaperones. And so how can you tell if there's a directionality or a cycle? So you cannot tell this if you have, let's say, HSP90 in one vial and another component in another vial, and you pour them together, and then you see a relaxation. Yeah? Because this is a non-equilibrium experiment. Yeah? You see the relaxation to equilibrium or to something like steady state or steady state turnover. But you have to measure at this steady state turnover to see if there is a cycle. And yeah, I forgot to mention here, you can use also many other of these softwares, and Markus Götz and Sonja Schmidt are conducting or did a comparison study of many of this hidden Markov-based analysis, and they would all give you kind of similar results. Yeah, if you give them yeah, if you tell them a certain number of states and you give them some criteria. Yeah, so this is not a problem of SMACs that you need to put the model in, but all these kinds of models. Okay, but now to the flux. So if you now want to know if there is a directionality, let's say HSP90 is in the open state, something binds, then it goes to the closed state, something unbinds, and it goes to the open state again. What you then would see, so then you would have all the forward rates and all the backward rates. This we could determine if we assume the model is right. And if you then take the product of these forward and backward rates, the logarithm of this, and multiply it by the thermal energy, then you get something like the free energy in this cycle. And this would be 0 if there is no energy source or no flux, and it would be larger than 0 if there's a flux. So for this, you need the rates at steady state. And this is what I meant before. So you cannot have two whiles and pour them together. This is not the rates. You need the rates while the system is in steady state. And I think this you can only do with single molecule experiments. If the system is not completely simple. Good. So now we calculated this, of course, for all the systems. And I mean, for HSP90 on its own, it's 0. This is nice. Then for HSP90, CDC37 T11, 0.4. This is also nice. Yeah, this gives you a little bit of feeling for the error, because yeah, I mean, there's no energy source, so it has to be 0. But then, unfortunately, if we add ATP, we get kind of the same result. So it really, I mean, what we can say, if this model is correct, that there's no real directionality even in this system. And yeah, why is this a result I mention here? Because there are several models published like this. Yeah, they all look a little bit like this here. So you have HSP90 in a state, or let's start here. You have the kinase, it recruits CDC37, then it binds to HSP90 in the open state, then it closes, then CDC37 is released. There's also some phosphorylation going on in the meanwhile. Then the kinase is activated, then the kinase is released, and then you are back to the open state. And if you now have these 4 states, yeah, let's say HSP90 on its own, HSP90 open, bound open, closed bound, and closed, only one molecule bound, you would need to see a directionality here. Yeah, and we don't see this. So this is really unlikely. But it's not really so satisfying, yeah, just to say, okay, this is unlikely, yeah, because I mean, we have here now one model, okay, you can use another model, maybe where there are 6 states, and we could use 6 states, and you would also not see a directionality. But how can we do better? And this is tricky with these kind of models, and more color FRET can or helps us there a little bit to need less of a model. And this is what I try to show you now in the maybe next 15 minutes or so. So what can we do with multicolor FRET? So what we have now in the setup, yeah, and please interrupt me if I'm going too fast. So now we basically added another camera. Yeah, you could also use 4 cameras to have now 4 colors separated. Other than that, everything's the same. Excitation and total internal reflections, polymer bound to the surface. And I mean, now you need more proteins in the background, so it gets a little bit more messy. But yeah, with the third protein, it kind of works okay. So we ask ourselves now, is there a conformational cycle with ATP and the co-chaperone? Yeah, we don't see it if we assume this model, but can we do a little bit better? And so for this, we had a very detailed look at this system, HSP90, ATP, and the co-chaperone P23, which is SBA1 in yeast. So we are working with the yeast system here. And first started with this 3-color system, and only excited the green dye here. And with this, then you get kind of 2 FRET efficiencies, one green to orange and one green to red. But with this, we can already tell a little bit about ATP binding, and we get 2 kinds of traces. So one is this, and this actually tells you that ATP binds to the open state. So the black trace tells you how close are the green and the orange dye, and they are far apart from each other. in this time trace. So it's the open state. And the blue trace tells you if ATP is bound or not. And you see ATP bound, unbound, bound, unbound, bound. Then we have this second kind of traces. So here you now see, let's start here, black trace high, so closed state. And then you see ATP binding. So here now ATP is bind low, then ATP binds, some energy is transferred. So you see now FRET basically from one die to the other, and then ATP unbinds. So we see now that ATP binds to the open and the closed state. And yeah, unfortunately, there's no direct coupling to this open-close movement. It's not like HSP9 is open, ATP binds, it's closed, it's hydrolyzed, and it opens again. So not like myosin or kinesin, where this open or this stepping is strongly coupled to the movement, to the conformational change. So then, yeah, we ask, okay, maybe in the presence of P23, there could be some directionality. The problem is now you don't want to have many fluorophores in the background because this spoils your signal. And therefore, we try to get vesicles, 100 nanometer diameter, where we include these molecules. Yeah, and I mean, this is the ideal case. So it would be in most of the molecules, there is no molecule, and many there is one molecule, and some two, and in very few ones there's all three components. So HSP90, ATP, and P23. Actually, we made some pores in there that ATP can come in there. This actually was a very nice system, worked very well. Unfortunately, really all the clients we tested, they all bind somewhere to the vesicles or so. So we could not do a single experiment with a client like this. So this is unfortunately. This only works for very few systems, but if it works, it's really nice because you have a concentration of about 1 micromolar in here with only having one molecule. Now you have 3 molecules, but each of it would be 1 micromolar. Okay, and what we can do now, and this is really independent from a model, you just count how often does a molecule transition from one state to another state. And we also have to define states here, but I mean, you can just define 3, in this case, arbitrary states here. And if you see a cycle, so a directionality in these 3 states, you have a coupling in this system. Yeah, you don't have described the full system yet, but I mean, you have seen this then. So basically, what you can see here, so this is currently just excited with the green laser, and you see here energy transfer all the way from the green to the orange to this far red dye, ATO647. So you see ATP is bound here. Then you see here a transfer to this, mainly to the red dye. So this tells you P23 is bound. And here you see, no transfer to any of the red dyes, but to this orange dye. So here HSP90 is closed. And then you see here P23 binding again. So we cannot distinguish this trace from this trace here in this, just by looking at the signal. So therefore, we have to put these 2 kind of together here. But 3 states is enough to talk about directionality. I mean, 2, it doesn't make sense, but for 3 states, this is okay. And now you can ask just how often do you get the transition from 1 to 3, from 2 to 3, 3, 1, and 3 to 2. And we clearly see differences. If you see 1 to 3, 11%, 3 to 1, 70%. We have a quite big error, but I mean, still it's significant. So basically what we can say that there is some directionality in these 3 states that we really kind of randomly picked off the probably tens of states this system can take, but I mean, this is what we did. And now you can calculate this free energy change. And you, instead of the rates, you put in here just probabilities. You can take exactly the same equation, just because we look here at single molecules, you put the probabilities in there. So these are all the forward probabilities, these are all the backward probabilities, and we see that we get something like 3 kT. yeah, this was not fully what we wished, yeah, because from I don't have this here, from ATP hydrolysis you would expect to get something like 20 kVt. So it's still weak coupling, but it looks like that there is a little bit of coupling in this system to the ATP hydrolysis. But I can tell you now, even 8 years later, we still don't know what the ATP hydrolysis in HSP90 really is for. Yeah. It is there, it has to be able to hydrolyze, yeah, a part maybe goes in here, but not much. Okay, I think I skip this and go to this part. So this was a little bit, yeah, so it can help us to get the succession of states, 3-color. Then you can also ask about coordinated action, yeah, also cooperativity. So what we ask here is, if we have 3 components in the system, yeah, how do they work together? And for this now, it's good or helpful to have this alternating laser excitation. So we didn't do this in the previous study, but here we have now here the green and blue excitation, because this helps you to better define the states. Yeah, before I kind of randomly picked the states, but now we want to define them. Why can this be better in defining states? So if you only have 2 colors, then you have a, let's say, high FRET, low FRET state. If you're lucky, you have an intermediate FRET state, but that's it. Because you only have this kind of 1 FRET efficiency, 2 signals, donor and acceptor. But if you look now at this, you have now 5 signals, or with the control, even 6 signals. Yeah, if you also excite the red one. And because from the blue excitation, you get basically then the blue, the green, and the red signal. And from the green excitation, you get the green and the red signal. And from these 5 signals, you can calculate 3 kind of FRET efficiencies. Yeah, we call them partial fluorescence, because they are not exactly the FRET efficiencies, and it's a little bit more complicated to get real distances out of this. But it's yeah, it's related to the FRET efficiency. And now you have this 3-dimensional, yeah, cube, you can say, where you can put the partial fluorescence in the 3 dimensions, and now you can define certain states here in this 3-dimensional state. And for example, I mean, if you would look at the projection down to, or up, it's written up here, to these 2 partial fluorescence, yeah, you could not distinguish these 2 states. But if you have all 3 partial fluorescence, you can distinguish states that otherwise would be not distinguishable. And so we have here now these 4 states well defined, so open state of HSP90, closed state, and both of them with labeled nucleotide bound, yeah, also the open and the closed state. So we cannot say if this labeled nucleotide is bound to the left or to the right one. In principle, you could say, but this was a little bit there, the states were not so clear. But we have these 4 states, and now, yeah, this is a labeled non-hydrolyzable nucleotide. Good, and now we wanted to see a correlation or cooperativity of this ATP analog and a co-chaperone AHA1, because it was known that this AHA1 accelerates the ATPase of HSP90 10 to 15-fold, and actually the only co-chaperone that does this. And so we looked now at all the transitions, so just in the presence of NP-PNP, and then with ATP and with AHA1 and with both of them. And now we only look at these first 2 transitions. They are the most interesting one. And I think it's nice to show the power of this 3-color FRET. I think this you could not get out of 2-color. So this is the original rate. So then you add ATP. So you see this rate is accelerated. And then you add AHA1, you see it's also accelerated. And if you both add both of them, you get much more. So here, kind of AHA1 accelerates or amplifies what ATP is doing. Here you see the different, the opposite thing. Yeah, you see here that ATP has a kind of inhibiting effect. So this rate is slower with ATP. But then if you add AHA1, this is kind of neutralized. So AHA1 multiplies one effect of ATP and inhibits the other one. Or if you would put this into a picture, yeah, you see these transitions. So these are the two rates you would have seen here. And you see ATP accelerates this rate, and AHA1 further accelerates this, while ATP decelerates this rate, and this is inhibited by AHA1. So you see here can decide if this is cooperative or if it supports each other, or if it does not do that. Okay, and then the last example, it's the one that has the least progress, but I think, I would just like to show it, and we are still working on it. So what we now would like to see is the interaction with three proteins. Maybe you have observed before, we always this kind of trick. One of the die or one of the component was the ATP. This is always easier. And then we had two proteins. So now we would like to have three proteins. And this is still tricky because dyes on proteins, they don't behave as well as they do as nucleic acids. And so it's very hard to get traces where there are really all three dyes there, especially as you cannot have high concentrations of these molecules in the background. So we are trying now to make these constructs where we have a leash and a molecule there. We actually also have this construct here. I'm just trying to measure this because you will see it's kind of tricky to get the trace with this system. We thought we can work with one protein in the background. We are still trying to do this with starting now with zero mode waveguides from Jerome Weninger. We get this, but we will see. I mean, this is the tricky part about the three dyes. You need to have them all three somehow together in your spot that you are measuring. We managed to do this. This is one of the first traces we got, but you see, I mean, there's still some way to go, still some work. But I mean, what you can see, just to see that the three dyes there. So the blue trace basically is blue direct excitation, and you can see that the blue dye is here, and here's the kind of the step. It's not a great step, but it's okay. So the blue dye is there. The green one was very nice. It's there. Each is there. And also this reddish dye or far red dye is also there. So you see a signal there from this dye actually pretty long. With the FRET, it's a little bit tricky. So we see a clear FRET here between the HSP90 and the CDC37, with the nice bleaching here. The other two FRET, I mean, maybe you also see a little bit here FRET from the CDC37 to the AHA1, but it's really tricky. So we really have now some hope that the zero mode waveguides will help us. But I mean, you can start now, and I mean, this is now the power of the thing, and that's why we put a lot of energy in there. You could now tell how this protein, one of the other, bind HSP90, and then are released, and then even confirmation of the change of HSP90, what you don't see here because it bleaches then. But you could now see really the succession of the states. And I think it's, from all the data, also some things I showed you, I believe that it's not this determined succession of states, what many biologists like, that you have one state and then one protein binds and then the next and then a next one and something happens and then they go apart again. So I think this is much more random, and of course this makes it much more difficult to detect. The proteins just bump into each other, and at some point they are in a correct state and they form a complex that can then do something, and then they probably are released again. But so this is ongoing work, and just should give you a little bit impression of what can be done. Good. Now, just a few minutes. Tim also told you a little bit about structure determination. So one can use now single molecule FRET to determine structures. And not only fixed structures, but also structural ensembles. And I mean, this is the real strength. You can look at these molecules in vitro and see then also how solid they are, kind of. For this, 2-color FRET is completely sufficient, but you have to do many experiments. Yeah, so you basically, in this case, we did more than 150 2-color FRET experiments with dyes in different positions, and then you can get something like, in this case, the open structure of HSP90. Yeah, and for this you use a different kind of setup, or I mean, you could do it with turf, but it's just easier to do it with confocal FRET. I think maybe Jens would argue different. I don't know if he's here, but it's fine. You can do it with both. Yeah. But as we did it with confocal, I just would like to explain to you. So what you have now is the molecules free floating. They float through a confocal volume. The disadvantage is you only see them for 1 or 2 milliseconds, so you cannot look at the succession of states anymore. But this we are not interested here. Here we are interested in the structure. So therefore, it's enough to just see the snapshots of the molecule in different states. And from this, you can then get, so you get the FRET efficiency, just snapshot of this. From this, you can get distance distributions. Yeah, and getting distance distributions was pioneered by Jens Michaelis and Klaus Seidel to get then real distances out of that. And first we used this then and said, okay, we measured really many distances. I think it's still at least the most published distances on one molecule, or on a dimer it is here. So it's 144 distances we measured, and for each of them you get this distribution here. And you see kind of 2 states, the open and the closed state. From before, you know that in the closed state you still have 2 states, and in the open also, but this will then contribute to the variance, basically to the width of this state. And you can already see here a little bit, so this would be in the presence of ATP, here AMP, PMP, where you mainly get the closed state, and it's also a little bit more close, it's more or less shot noise limited, while in the open state it's much wider, so you see a lot of fluctuations there. But so we can now assign these 2 states, and for each of the states we can then make a distance network, and get then something like this. So this is the closed state, what you see in the solid blue, and then you can get something like 1 sigma and 2 sigma structure. So basically from the width of this, you can say, okay, I get the structure from this and from this FRET efficiency, and this kind of tells you a little bit how strong are the fluctuations in this state. So here I would call them kind of fluctuations, but here, I mean, I also showed them kind of fluctuations, but we cannot tell, this could also be several states, just very quick transitions between those states. And at some point, it's also philosophical if you talk about states, or if you say this is fluctuations. So, but you see that this is way more dynamic in the open and less dynamic in the closed state. And the nice thing is now, if you look into the structural ensemble that we got, which also put a lot of confidence for us in this, is if you compare this now with open structures from HSP90 homologs. So for the yeast HSP90, there is no open structure, but this is for the E. coli HSP90. It's called HTPG. There is an open structure, and this basically superimposes very well with the 1 sigma structure, especially this CM here. The end domain is a little bit differently aligned. Another homolog is a GRP94, and this you can see here. And this also nicely superimposed, in this case, with 2 sigma structure. So in this structural ensemble, we have several other structures in there, which gave us kind of confidence that we saw something real here. Good. And finally, what Tim just mentioned, we did then this study to really figure out what is then the precision that we can measure the distances in such a network. I mean, this was the question, and Tim and I discussed a lot about this, and we still don't know. Actually, if some theoreticians listen, maybe you can have it, but we have the first step for this, namely we can tell how exactly can we measure one distance. In a network, you will be better, yeah, because you have more information on in this network, but yeah, it's still unclear what you really can measure. But yeah, what we did, we had many labs, and we could see that even a single distance within DNA, you can measure with angstrom precision. And so this gave us confidence to move on. And now there is a new study, now means since several years, which tells you it's not so easy, spearheaded by many people. I think especially Don Lamb and Torben Cordes, but also Klaus Seidel, Philipp Tinnefeld, but also Tim and Miele Hendricks, Achilles Kapanidis, Jens Michaelis, Michael Schlierf, and myself. Yeah, we all try to contribute our part to get some distances there. And yeah, I just heard last year that we are close. So yeah, we will see. So stay tuned for how it is with proteins. So it's a little bit more tricky. But in principle, this just means you have to measure more distances. Yeah. Okay, so this is the final good news about the distance measurements in single molecule FRET, but it's still hard and there's not so many structures measured yet. Good, and now I think I have 2, 3, 4 minutes left. And just very quickly, another way where I think single molecule FRET will be very powerful. Some of you might have heard about liquid-liquid phase separation. So some biologists believe that this explains all the shortcomings we have in explaining how proteins interact and regulate themselves in cells. So a big puzzle is, how can functional complexes be formed in a cell if all the affinities are so low? Yeah, and so then the idea is to have compartments. And of course, every one of you knows that there are compartments in cells. You have the cell membrane, you have a membrane around the nucleus, but this is not enough. You would need other compartments to get higher concentrations of some molecules. And several of them were known before, but since a few years, this is really a hot topic in biology to look at phase-separated parts without membrane. So they are also called membraneless organelles. They are called clusters, they are called droplets, they are called condensates. Punctae is also a name. So it's many different names, but in the principle is that you have some phase separation where you have then a higher concentration of something. And this is very interesting also point of view, because it goes across several length scales. So you see these, I call them condensates now, with these bigger clusters on a scale of micrometer. And this is what most people see and investigate and try to figure out what interactions are there. But then, I mean, if you look close, and then there is also the interactions on the nanoscale. And also, I mean, to get from, let's say, 2 proteins interaction to these big clusters, yeah, they're also there. You need to have some nanoscale information. And for this, single molecule fluorescence and FRET is very good. So this was the question that we started this year. So how does this cluster formation start? How is it regulated? Is this irreversible? So how does this work? And our collaboration partner, so this is together, I forgot to mention, with Ritwik Sabakar and Alex Reinhardt, now in Cambridge. Both of them, Ritwik was here at the Max Planck in Freiburg. And he had seen that such clusters are forming, such big clusters are forming of this protein. It's called negative elongation factor. And yeah, it's important for stress response and thus actually transcriptional downregulation. And yeah, this has been seen before. There's also some idea how this works. It's a phosphorylation combined with the interaction with intrinsic disordered regions. But this is kind of what they analyze, the final state. Yeah, now we're interested, I mean, how do you get there? And for this, we first had to go to way lower concentrations. And we first looked then with a little higher resolution. So this is an Airy scan image, and this is unstressed cells, this is stressed cells. And what you can see many of the small clusters in both of the cells, but one big one here. I know I started a little too early, but you see one of this bigger cluster in these stressed cells. So this is NELF GFP. With DAPI, the DNA is stained, and here the nuclear lamina is also stained, but we figured this would not be important because the DAPI stain works very well in the additional images. Okay, the problem is, I mean, it still takes quite some time to get this image, so you don't have a very good time resolution. So therefore, we went to a little different microscopy, namely hylomicroscopy. So it's kind of an optical sheet illumination. And yeah, so what we imaged, I mean, this is kind of 10 cells now. We go at much lower concentrations than many other labs go. It's not super resolution, but it's kind of, because we have low concentrations and therefore you can localize things kind of well. And what you see now is these small clusters, and they are really dynamic. They are moving. We can track all of them. We can determine their size over time, what we do. And then you see, so here's the first cell, but then this big condensate forms. Yeah, here also. This one is kind of then the last one. Yeah, and in the other cells also. So these are basically the cells now where we saw this a clear condensate formation, and we analyze this. Now I show you one little piece of the analysis in 2 slides. But what was very surprising, I mean, some people saw these smaller clusters, but then they always said, we see the small clusters and big clusters, and now analyze the big clusters. So I think this is now, yeah, we try and others try to analyze them, but just starting now. And why do I think this is so important? Because then we looked at unstressed cells, yeah, where you never see this big condensate forming, and you also see these smaller clusters, and they are also very dynamic, both within, so they move around and they also change their size. Yeah, and with this, I think that these dynamic transient clusters are actually also very important. Yeah, so E'(CC) call them transient clusters. And yeah, so now we try to analyze them and find differences between these clusters and under different conditions, yeah, in stressed cells, in unstressed cells. So these are basically, each of this is a cluster. And then you look over time, and the size is coded with these colors, and you see that there are size changes. Yeah, there is some differences there. So we can get the dynamics of the clusters, both position and size. And we can also get the size that is necessary for nucleation when there is irreversible growth. And actually, it's really many molecules. But the 2 or 3 other studies I'm aware of where they try to determine the size of the nucleation for these big condensates is really, they also have close to 1,000 molecules. And this is what we puzzled all the time. And that's why I focus now on the small clusters, because these big condensates, yeah, there is thousands of molecules just of one kind, but there's also other kinds. So it's 5,000–10,000 of molecules. And yeah, I somehow think maybe the smaller clusters having 50 or 100 proteins are more relevant, but I have no proof for this yet. It's just my personal opinion. Okay, but now I think my team says 50 minutes, so I think this is enough to leave some space for questions. And yeah, I hope I could show you that the future for single molecule FRET looks bright. And yeah, I mentioned my collaboration partners throughout the talk. And yeah, I thank all people from my group for all the work they did, these institutions for funding, and you for your attention. Thank you.
It always sounds horrible, one person clapping, I think, but, but that was, I'm sure others are sharing with me in thanking Thorsten for a fantastic talk. Are there any, if there are any questions, if people would like to just, if they have a question, ask, just to say you have a question in the chat, and then we can go to those specific questions. We had a couple of pre-registered questions, and I noticed those people are there. So Susan, did you want to ask yours maybe to start with? If you want to unmute and ask your question. That was Susan Zelga, sorry. Susan, are you there? You were a minute ago.
No, sorry. Hi, I'm here. Hi.
Oh,
I'm
brilliant.
sorry. Oh, sorry, I forgot actually which question I was uploading.
That's all right. I've got it here, and it's similar to Nicholas Vickers, in fact. So both of you are interested in what the biggest challenge to the 3-color FRET measurements were, and for Nicholas, especially in terms of the dynamics. So, Thorsten, could you maybe comment on what are the biggest challenges to doing a 3-color experiment?
Yeah. So a little bit of work is to get all the correction factors right, but this is published and can be done. And the real challenge is to get the 3 dyes long enough. Yeah. So the problem is, so they are not really exponentially distributed. How long a dye lives until bleaching, but kind of. So basically, 1 dye for 100 seconds. This is okay, nice. 2 dyes for 20 seconds is also okay. 3 dyes for 20 seconds is, you have to look very long. It's just a probability that all of the 3 dyes are good. And this is the challenge, because if you really want to look at dynamics, you need all 3 dyes for, yeah, it would be good to have at least some traces more than 10 seconds or something like this. And this is hard.
Brilliant, thanks.
These better dyes, yeah, it's kind of a little bit easier on nucleic acids because the dyes are a little bit better there. But I mean, so Chip also just published something months ago or so, and even on nucleic acids, he probably also would have loved to have nicer traces. Yeah, it's really tough to interpret this trace system.
Brilliant. Abigail, did you want to ask your question?
Yeah, sure. So I was just wondering about the cluster of observations that you made and how easy you think you could adapt the assay so that you could sort of analyze the exchange between neighboring clusters, if that was happening.
So for the live cell part, yeah. Yeah. So yeah, I forgot. So I have it written here. So co-phase separation. So of course the interesting thing is, and we have first data, but of course we also work with HSP90 there and HSP90 interacts with NELF. But I cannot show this here at the moment. But this is the interesting point. So if you have co-phase separation between 2 parts, and if they first maybe do phase separation on their own and then get together, or if one recruits the other one into a phase separated domain, yeah, and to see exchange. But this will be the hardest part. I mean, already to tell which is the, they call it driver of the phase separation. And yeah, this is the real cool stuff. And for this FRET, of course, it's great. So therefore, yeah, we want to do FRET. But again, the problem here is also not the technical thing. Yeah, I mean, you have to do some controls and things like this, but the problem is to get the right concentration of these 2 dyes. So it's already hard to get the right concentration of 1 dye, because if the concentration is too high, your background is too high, you see nice cluster condensate formation, but not the small clusters. If it's too low, you don't have enough molecules to form the big clusters. Yeah, so you have to be at the right concentration, and every cell is different. And if you now have to get this for 2 colors, it's more tricky. That's why we start also do injecting. Yeah, so basically, so this was the express dye, and then you can inject the second dye. But yeah, it's a different kind of experiment, but I think with really high potential.
Brilliant. Virginia, would you like to ask your question? I can ask it otherwise. Great talk. I have a question. Is there any option of combining smFRET and FLIM?
Yes, you could combine it. I think maybe you can have, I mean, I'm sure this has been done even, but now at the moment, I cannot just tell you which publication. Yeah.
So, certainly FLIM-FRET is definitely a thing, even if I can't say it. So yes, that's certainly been done before. Thorsten, I think that's probably the most of the questions. I've got some lovely things to discuss again later with you. But I want to just thank you again for an absolutely brilliant and far overarching talk there from 3 colors to dynamic structure determination all the way through to what we would commonly call in my lab blobs and live cell imaging of condensate formation. I think this is a real testament to the power of all of these types of methods. So thank you again for that, and sorry it was a rush for you to get here on the train. And thanks again to everybody for joining. We'll be in touch via email and Twitter and LinkedIn for the next exciting seminar in a couple of months' time. And if anyone would like to think about using a flex to do any of the experiments that Thorsten's talked about, especially the single molecule confocal ones, that's what we're geared up to do right now. So do be in touch for that. But otherwise, thank you again, Thorsten. Thank you everyone for joining, and we look forward to seeing you all in a couple of months' time.
Thank you.
Cheers, Thorsten. Thanks ever so much. Really appreciate it.
Thank you.
We'll talk again very soon, I hope.
Yes.